Welcome to the Virulent Vortex, a podcast in which we discuss infectious disease across scales, from pathogens and molecules to populations and pandemics and everything in between. My name is Jaap de Rode. I'm a professor of biology here at Emory University. And today I am joined by Anice Lowen, who is a professor of microbiology and immunology, also here at Emory. Anice, welcome to the show.
Thanks so much, Jaap. Happy to be here.
Now, you study influenza. That is really kind of the thing that you really like studying. And you specifically like studying co-infection of influenza. So tell us what that is and what you find interesting about it.
Absolutely. So yes, we've long been interested in what happens when two viruses come together in the same host and in the same cell. Okay. And early on, we found a very interesting effect where it was clear that two viruses in the same cell are helping each other. Okay. So very often, if one virus infects a cell, it will fail. It'll fail to make more viruses, which is, of course, the main goal of a virus, is to copy itself and make progeny. But if multiple viruses enter the same cell, then the likelihood of success are greatly increased.
Okay.
So we're really interested in this beneficial interaction that we see in co-infected cells.
Right. Okay, so maybe just take a step back. So you said that these influenza viruses are actually pretty poor, pretty bad. So if you have a single one going into a cell, it basically fails. Is that because it cannot get all its parts together and create new ones?
Yes. So all the reasons why it fails, we don't fully understand, but you're right. The virus appears to be quite inefficient. It's interesting that you refer to lots of different parts, because the influenza virus genome is made up of multiple pieces of RNA. Okay, there are 8 pieces of RNA that comprise the genome. And we do see that one reason why an infection can fail is that only a subset of those 8 segments of RNA get replicated and expressed. Okay?
Right.
So you have sort of an incomplete infection, if you will.
Yeah.
And that problem, of course, can be fixed if you have multiple genomes that are delivered to the same cell. That is a simple numbers problem. You have more chances to get the right set of 8 segments present and functional.
How does a cell get multiple viruses?
So multiple viruses can enter the same cell quite simply if multiple viruses attach at the same time and are taken up by the cell at approximately the same time. And we know that in a natural infection scenario, that's actually quite likely to happen frequently because viruses spread in localized areas, so sort of patches of infection where the viruses would be quite concentrated and likely to have multiple individual viruses attaching to the same cell.
And that is within the lungs, for example?
Yeah, so
those areas?
So for influenza viruses, this would be in the respiratory tract, and including in the lungs. And of course, that's a big tissue. You could think about how a virus population could be quite widely distributed, and co-infection would appear to be unlikely. But because of this patchiness, the spatial structure that goes on during infection, it really increases the chances of co-infection occurring.
Yeah. So say if I had flu and I were to sneeze and virus particles would make it into your lungs, because that's how it works. Not very pleasant to think about, but that's how it works.
Yes.
So if there was only one viral particle making it to you, you wouldn't get an infection. But if I give you enough, there is enough chances of different viruses to get together to the same cells and then start that replication cycle.
Yes. So the transmission stage, that very early stage of infection, is really interesting to think about when we consider this benefit of multiple infection that I've described, right? Because if just one virus transmits sort of individually with that sneeze event, then I would predict that it is very likely to fail and transmission actually won't be productive. But if sort of many viruses transmit individually and each infects individually, then maybe you get the jackpot event, but 1 in 100 would actually initiate the infection, So that can happen with a single virus. Conversely, you could have a situation with the sneeze where there's multiple viruses in each drop of spittle that's flying through the air, And so you could get a coordinated deposition of virus onto one cell, even in the initial transmission event.
And so you have these co-infections. So each virus has these 8 genetic elements, basically these portions of their DNA or RNA virus. And so if those viruses are the same, you end up with the same virus. But I can imagine that those viruses are completely different. They could come from different individuals. They could even come from different species. And is that actually how you get these completely new viruses? Because they combine these different bits of their genome?
Yes, so that's an excellent question. So it's an important feature of influenza viruses, and due to the fact that their genomes have these 8 segments, when they co-infect, the segments can swap back and forth. Mm-hmm. Okay, so we call it reassortment. Yeah. And you can have progeny viruses that have chimeric genomes coming out. So it's an important source of viral genetic diversity. Yeah. And it's particularly important in epidemiological terms in that it's where influenza pandemics come from. Okay, when we have this genetic exchange happening between a virus that's well adapted to humans and a virus that is well adapted to some other animal host, the new chimeric form that comes out can have pandemic potential. Yeah. And we've seen that repeatedly over the decades.
All right, so you have talked a lot about these viruses co-infecting individual cells. But when we think about the body, the human body is very, very many cells, right? And so at the level of the individual, the host, the patient, how does that co-infection play out at that level?
Yeah, so I'm glad you asked, because this is something we tackled pretty recently, try and sort of zoom out from the individual cell and think about whether these beneficial interactions that we've observed actually determine infection outcomes when we think about the whole host. And in particular with this question, we were thinking about the rare but important scenario where you have infection of a single individual with 2 distinct influenza viruses. So it could be a human that's infected with a normal seasonal influenza, and then maybe an influenza virus that's actually well adapted to birds. Okay. So that bird virus normally would fail in a human. It wouldn't replicate well, it would make few progeny. But we hypothesized that in the situation where it's co-infecting with a human virus, maybe the human virus would help the bird virus to replicate and thrive in that new host. And of course, the hypothesis was based on the strong evidence we have that there's beneficial virus-virus interactions happening within cells. But when we considered the virus populations in a co-infected host experimentally, what we found was actually a very opposite dynamic. We saw that the 2 viruses co-infecting the same animal competed with each other, such that one virus would prevail and suppress the replication of the other one. So that was really puzzling at first, but with the benefit of more experiments and hindsight, it does actually make perfect sense. Okay, so within a cell, viruses are helping each other. It's a beneficial interaction. But outside of cells, viruses are looking to infect a fresh new target cell, and they're competing with each other to find those fresh cells. Right? And so that happens because a virus cannot infect a dead cell. It also can't infect a cell that's already been taken over by a virus, and if the virus has been converted if the cell has been converted to a virus production factory, then the machinery that the late coming virus needs is sort of already taken, and it can't be used. Okay, so we have this strong competitive effect when we think about viruses moving between cells.
Oh, interesting.
Yes, it is interesting. And so it's really important because, as I mentioned, there's a sort of rare situation where you can have co-infection between 2 quite distinct viruses. And there, if the bird virus is able to copy itself within the human, it's very concerning, because that process would give the opportunity for the bird virus to evolve and adapt to the new human host, which then creates pandemic.
What have we learned that we could apply to the future and try to prevent these pandemics from happening?
Yeah. So I think the biggest sort of practical information that comes out of our work on co-infection and reassortment is that to prevent pandemics, we should prevent spillover and co-infection, okay, and the non-word transmission from the co-infection. And so that's not easy to do, but we can think about where spillovers occur, okay? So again, we're thinking about the natural hosts of influenza A viruses that are not human, okay? They're the source of pandemic viruses. Yeah. So where do humans meet those species? And can we sort of limit those interactions between the non-human species and humans? Okay, so the interfaces are in agricultural settings, and live bird markets, agricultural shows, things like this, where you have humans interacting with pigs, chickens, ducks, and so forth. They have influenza viruses that are presenting a pandemic risk. Yeah. So, if we could limit the interfaces or at least surveil the interfaces so we know what's there and we can better prepare for emergence events.
So really preventing those co-infections as a starting point.
Right. Yeah. No, I mean, a great thing would be to, as a policy that could be developed up this to, insist on vaccination for swine workers and in agricultural settings, for example. They need to be protected against seasonal influenza so that you wouldn't have the situation where they would get both seasonal influenza and swine influenza at the same time.
Yeah. And that is not a practice right now?
No.
Wow. It seems like a logical thing to do.
Yeah. Requiring vaccination is not easy in this country.
That is a good point. Yeah. Well, Anice, it was welcome to have you here and to learn about your research on the co-infections in influenza. We hope you enjoyed this episode and we look forward to seeing you again. Thank you for watching.